panel of witnesses. This is the first oral evidence session in our inquiry into regenerative medicine. I would like to remind all members of the Select Committee to declare relevant interests before they first speak in the inquiry, and the declared interests have been published in the public information sheet. And I would like to remind all of those in the room that the meeting is being webcast, and therefore sotto voce aside will be broadcast, and the broadcasting system often remains on after the end of the session, so be alert to that. In the first of the two witness sessions that we're holding today, we're very much interested in seeking your views on matters relating to the quality of the science base in this area of regenerative medicine in the UK, and how that science might be applied to treating clinical conditions. And you've seen some questions that we wish to explore with you. Before we kick off with the questions, I'd like to invite each of our witnesses to introduce themselves for the record, and if there's anything you want to say as a preliminary, please feel free to do so, but keep your comments brief, because we have quite a lot to get through in the next hour or so. So perhaps if I could start with Dr. Vallier. If you just introduce yourself.
Yeah, thank you. I'm Ludovic Vallier. I'm a reader in stem cells and regenerative medicine at the University of Cambridge, and also director of the NIHR-BRC IPS Alcohol Facility.
Charles French Constant. I'm director of the MRC Centre for Regenerative Medicine in Edinburgh.
I'm Stephen Sachs. I'm director of the MRC Centre for Transplantation, which is at King's College London.
I'm Michael Linden. I'm a professor for virology at King's College London and the director of the Gene Therapy Consortium at UCL in London.
Okay, thank you very much. Well, perhaps I could kick off by asking each of you in turn to comment on how the UK ranks internationally in research in regenerative medicine, and particularly what you think are the strengths and weaknesses of the UK compared with other countries. Perhaps Professor Linden, you could kick off, and then I'll work along the row.
Yeah, so first I'd like to qualify that what I have to say is probably relevant to gene therapy in particular, which is gone through some developments through which other areas of regenerative medicines probably will go through. I've spent a considerable time of my career in the United States and have somewhat of a comparison in my area between these, and I must say that the potential for this area of gene therapy here in the UK is extraordinary. That's based on several aspects, I think, that can be considered. For example, the NHS, which makes it easier to translate clinical studies rather than in a private environment where different sorts of funding needs to be found. And probably even more importantly, the per capita impact that UK scientists have compared worldwide. So UK science, biomedical science in particular, is very high in impact. When you take these two things together, I think there's a very fertile ground for the development of novel therapies, and in a certain sense a unique environment, I think, that should be and is being taken advantage of currently.
Thank you. Professor Sachs.
Well, my main interest is transplantation, and but beyond solid organ transplantation. And some of us where I work have been thinking very carefully about how to transplant cells and make them successful. And there are a number of models and actual examples in clinical practice, I would say, sort of experimental medicine. So I've seen what happens there, and it's quite interesting because the history of transplantation is exactly this that's happening with cell transplants at the moment. There's an initial wave of success, but then there are the limitations become apparent. And it's so obvious, always in retrospect, to know what sort of monitoring systems should have been in place to try and learn from the experience and build along the way. So it's terribly important to make sure that these systems for monitoring and interventions are there right at the beginning. And this, although I'm talking about cell transplantation of adult cells, the same thing I think would apply to some sort of stem cells or poorly differentiated cells as well. So I think that's really important. The potential, as I see it, sort of from the outside, is very great. The science in this country has always been terrific. The exploitation of it hasn't been so great. So what I feel is needed is a real quantum leap, which is major investment, and there are examples of how this has helped, for example, the human genome, and help us to be alive and competitive. From reading documents, I've got a flavour that we want to be as good as, but I think if we actually want to lead and dominate, which is a good starting point and a good target, then I think we have to do something really radical. And I know there's a lot of talk about networks, but I think there's an investment that needs to be explored beyond that, in having big centres where all the sort of facilities that we've thought about and heard about can be properly integrated.
Is that about more resource or about different deployment of the same envelope of resource?
Well, ultimately it's more, but it's an extension of what I've seen already proposed. I think networks and hubs are terrific, but there still needs to be some physicality of co-location to be a really tough competitor with the US.
So more than is going on, more concentration than is going on.
More concentration is going on, and a real magnet to pull people in as well.
Yeah. Professor Charles Constant.
The question was, how good are we and what are our strengths and weaknesses? So, I mean, how good are we? We have a reputation for being outstandingly good at developmental biology and stem cell biology. And we also have outstanding clinical academics, although I think that's quite patchy. To try to put some sort of metric on this, though, is quite difficult because citation rates tend to be quite historical. So what I did is I looked at the number of invited speakers to the major international meeting, which is the ISSCR meeting that's held every year. The first ISSCR meeting, there were 4 out of 45 UK speakers, and a decade later, in 2011, there were only 4 out of 109. Worryingly, many of the 4 were the same as the ones who'd spoken 10 years earlier. 2012, the meeting that's just happened, there were still only 10 UK speakers out of 157 talks. So I think we're perhaps being a little complacent if we think that our expertise in stem cell biology is, just leaving everyone else behind. We're clearly there with many of the Europeans and with Japan. The US are obviously the major players. Now, in terms of weaknesses, I would agree with comments of my colleagues. I think there's weaknesses of scale. The regenerative medicine projects need to be large, they need to be multi-component, they need to be highly organized, because this is a very long endeavor, and the rather fragmented and short-term funding arrangement in the UK doesn't lend itself to that at the present time. There needs to be a much greater degree of cohesion. I would agree that there needs to be more co-location, but there also needs to be more cohesion between the different groups. And part of the problem here is leadership. Academic leadership, by its very nature, is all about influence rather than power, and that often makes it quite difficult to corral people into a very business-orientated model, which regenerative medicine probably needs. So I think we need better leadership, both from pharma coming down into academia and also within the academic community. And finally, I think it's very important that we have long-term funding strategies. 3-year or even 5-year funding cycles are just too short for this endeavour, and I see that as a current weakness.
So just to come back on those figures you gave for invited speakers at the International Stem Cell Research Conference, just looking at those figures as you gave them, it looks as though, if anything, on that indicator we're slipping backwards. Is that too pessimistic?
No, I think it would be unwise to read too much into the variation, especially as the geography of the meeting inevitably reflects the balance of the invitees. But I think in the round, they show us that we're definitely not clearly number 2 behind the US.
Is it Sorry, Lord Brose.
Yeah, on this particular topic, I mean, if we had 10 papers out of 157, does the ISSCR include industrial sort of application papers as well as science? And if so, what was the balance of our 10?
The ISSCR does include a large number of sessions. I feel a little bit disconcerted with one of the ex-presidents sitting here, but there does certainly include industrial sessions, but I have not included them in my listing. I've simply selected the academic or plenary sessions, and there was relatively little industrial input into those sessions.
Lord Patel.
I wondered if Professor Charles French Constant would agree that using the sole metrics of how many speakers were at ISSCR international meeting is a strong enough metrics to suggest where the science in UK is.
I would completely agree. I think it's extremely difficult to find metrics, and those metrics should obviously be taken along with the well-established metrics of citation rates and things like that. My point was that citation rates do tend to be biased by what happened some years ago, and I was just trying to find a way to bring to the attention of the committee a current snapshot. But like any metric, there are many weaknesses, and I would not wish to overplay this.
Could I move on then to Dr. Vallier?
Thank you. So my field of expertise is really human embryonic stem cells and human-induced pluripotent stem cells, so my comment would be really focused on this aspect. I think clearly the UK in the top 3 countries in terms of basic research, clearly there in this field. The US are first because of the sheer size and resources that are available there. But I think we can't be really complacent because Japan is coming very strong, especially on IPS and translation. We have also other countries like France and Germany that are doing very well in terms of translation and bringing application to the clinics. So clearly excellent basic research in the UK based on the environment, long tradition of funding, culture also of research on stem cell and developmental biology, which have created a unique network of knowledge and expertise, which make really the UK one of the top leaders for basic research. The weakness are, in my opinion, are clearly the translation of this basic knowledge to a direct benefit to patients, which need to be reinforced, and especially in terms of, I think, transfer of this basic knowledge into a clinical product, and that's a global challenge. It's not unique to the UK. It's a global challenge for any country that is in this working in this field. But that's clearly where some needs are currently.
Lord Rees, and then I'm going to turn to Lord Patel.
Professor French Constant referred to the short-term nature of the funding, 3 to 5 years. Is that an MRC issue or Wellcome Trust or anything that we could make recommendations about?
Yes.
Yes. That's obviously an issue for the funders, the Wellcome Trust and the MRC, yes. I think it would be valuable to convince them that longer funding cycles for more ambitious regenerative programs would be of value.
We'll be able to ask them about that when they come to give evidence for us, to us. So I'd like to turn to Lord Patel.
Thank you, Lord Chairman. My question leads on very nicely from the first questions, which is related really, and I'd like, if possible, for you to answer in each of your individual expertise areas as to what regenerative treatments are currently available and in what diseases, and looking further ahead, in 5 or 10 years, which do you think are the areas likely further succeed in your own particular expertise areas?
So perhaps Dr. Vallier could go first on this occasion.
So, I mean, clearly for human embryonic stem cells and human iPS, we are at very early stage of clinical development. So we are, on the first in-man kind of clinical trial. So clearly the first clinical application currently in place is macular degeneration. And what is several trials also currently in development for cardiovascular disease and skin disorder, especially in France. And the next round of application will be definitely neurodegenerative disease, I think. In my particular interest is clearly liver disease. In the next 5 to 10 years, we hope to be able to do first clinical trial with human ES cells for liver disease. And there's also a very strong push for diabetes, type 1 diabetes, in terms of transplantation of beta cells generated from human embryonic stem cells. So there's a broad panel of application from human ES and human iPS. Clearly, human ES will come first because in terms of safety and validation, there have been already a lot of work done on those cells, and iPS still need more systematic studies. But what is important is that there's no one stem cells for one kind of clinic, for every clinical application. There will be a panel of stem cells for a diversity of clinical application. Human ES cells being probably the broader potential, but other stem cells will be extremely important in the therapeutical panels that will be available.
Can I ask if you think there are any stem cell therapies available now for any other diseases?
Yes, hematopoietic stem cells, for example. Limbal stem cells are going now, and mesenchymal stem cells are also starting to be widely used. So yes, there is clinical development application, and traditional one, since a long time.
Okay, so you asked what's currently available. The first thing I'd like to say is that there are almost no regenerative drugs available, which I think is a critical long-term goal, a medicine that one could actually take that would promote regeneration. There are, as we've heard, a number of cell-based therapies available, and I think it's very important to split them into two. There are the rather small number that are directly addressing the question of cell replacement, where you're directly replacing a damaged cell, like macular degeneration, as we heard about, and the hematopoietic stem cells, limbal stem cells. But then a lot of the mesenchymal stem cell trials that have been done, these are not cell replacement. This is just exploiting the happy properties of these cells in immunomodulation and in protecting other cells within the tissue. And I worry that that will limit their long-term use. Ultimately, it would be better, I think, to use a drug that had the same effect. Moving on to what the promise in 5 to 10 years, which I believe was the second part of your question. Obviously, I think cell therapies will become increasingly widely used, and I think there will also be the growth of a device industry that provides effectively devices where these can be prepared without requiring vastly expensive GMP facilities in which to grow them. I think that regenerative medicines, true regenerative medicines, will come through. There are targets that pharma are currently working on. So I think it's realistic in a 10-year timescale to think that they will come to the clinic. And there's one other thing I'd like to highlight, because I think it often gets forgotten. There's an enormous potential of cell-based platforms for drug screening and toxicology studies, using the technologies of regenerative medicine, using iPS cells as disease models or ES cells as a source to generate very large numbers of human cells. And the hope is that this will reduce the number of drugs that fail in late-stage trials.
Thanks. There are some interesting things going on. I think mesenchymal stem cells are, for transplantation, have produced astonishing first results. And let's see how well they can be extended. So I think that has a lot of mileage, and we'll see development in that area. There are other areas in which that can be exploited because, as you've heard, these cells were studied, and we've studied, other people have studied these, expecting them to trigger an immune response, a rejection response, but they do quite the opposite as stem cells. And so they are useful to suppress the immune system, and you may see this type of therapy coming in for other transplants as well to provide immune suppression. That's how they've succeeded in a particular problem with bone marrow transplantation. There are, I've mentioned, other people have mentioned islets and hepatocytes as well. They show this initial remarkable success, but people scratching their head at the end of 2 years wondering why they failed. So once again, they will develop, and there's no reason they shouldn't take a step change over 5 years, providing that the right sort of monitoring and interventions are available to deal with it right from the start. So there are interventions that I think ought to be, and I know in the broader definition of regenerative medicine what we're talking about, molecules that aid therapy, stem cell therapy and cell therapy, are really important. And I think those are there already. They haven't really been considered in this, the background literature. But it is possible to biologically modify stem cells, other cells like these things that we're talking about here with islets and hepatocytes, so that they'll get past the first stage of recognition by the host. And that's really important for all stem cells, is to be able to implant in particular numbers so that they overcome the inflammatory response, which in some cells is inevitable. And there are ways to manipulate cells now which can be broadly applied. And it's an interesting thing about transplantation, which I think will be seen in stem cell therapy as well, with the things that have been mentioned, that if you can actually get cells to embed at this very early stage, then it's very likely that that will be seen as a late impact. So in other words, if you just do something for the first 24 to 48 hours to a stem cell to make it more acceptable, then that benefit will be seen late on after the use of these stem cells, let alone having to tackle the immune response, because we don't know whether the immune response will be a problem. It certainly isn't with the cells you start out with. But as they differentiate and replace the tissues they're supposed to become, it may well be that then recognition of foreign tissue in the midst is more of a problem. So I sort of use that as a slight vehicle to talk about technologies that have to go alongside these real concrete examples of things that do exist, and hopefully will develop over the next 5 to 10 years.
Professor Linden.
Thank you. To answer your question directly, for frontline clinician, I don't think there is a therapy available that's based on gene therapy currently. But I think that needs to be seen in the context of where this field is. And if you look at the maturity of different therapy approaches, small molecule approaches are probably the most mature ones, the pills that we take every day, and then next one might be antibody-based treatments, and then cell-based treatments, and very immature in a certain extent is gene therapy at a certain point, if we're looking with respect to the application for a frontline clinician. But then we're looking at it from a scientific point of view, and there we have to say that there are very many and counting applications that are in a proof of principle stage in patients, early clinical phase studies, phase 1, phase 2 studies. Quite a few of the prominent ones actually coming out of the UK. For example, for haemophilia, a study that was published in the New England Journal of Medicine last year. Eye diseases have been published by Professor Robin Ali here in the UK. And not to be forgotten, Professor Adrian Thrasher's X-linked SCID immunodeficiency trial, all of which actually now show clinical benefit. And the question will be, I think it was very well pointed, because the question now, and maybe the challenge to this committee and many people, it will be to how can we make this proof of principle into a frontline therapy. And I think that probably will involve from funding bodies as an MRC and the two collaborative approaches with pharmaceuticals and small biotech companies. Collaborative approach to actually make this happen. I think, in my view, in the area of gene therapy, this is one of the challenges. Proof of principle is there. Proof of principle, the UK was leading together with the United States institutions. And now the question is, how can we make these into actual drugs? And I think there are many aspects that can be discussed in this area.
Did Lord Cunningham want to follow up?
There's been some criticism or some argument that in the UK our great strength is in basic discovery science rather than in translational science or in converting what we discover into practical applications. Now, that's not restricted to your disciplines. It's a general criticism of fundamental scientific research in this country. The government has produced its view on this, the BIS, and they outlined long-term actions for government to reduce regulatory burden, effectively fund and promote research. Do you agree with that, or have you different views about that? Or what are the problems? What do you identify as the problems which prevent this translation from the excellent, world-leading in many cases, basic research into practical application or into products from pharma or where else?
Okay, I'll begin. I'm sure there's much to be said after this. In the field of gene therapy, at least, and maybe it is restricted to this field, the development or the translation of bench research into clinical application is a continuum that employs a very complex continuum that employs in the beginning the researcher and at the end stage pharmaceutical industry. Now the point is, if some of the components in between are not in place and supported well, then the end stage will not even begin to engage. And in the field of gene therapy right now, and sometimes I act as a consultant to a pharmaceutical company, I see that and this, maybe you should ask these people as well. They're getting ready to get engaged in this field. For example, the number is out there that by 2015, the value of the gene therapy market could be globally as much as $300 million US dollars, and that's in the very early stages already. Yet these companies are positioning themselves into engaging, but are not quite ready yet. And I think the challenge will be to identify how to ensure a continuous development of any particular therapy from a promising proof of concept into a viable drug, which ultimately will involve very innovative pricing strategies. How do you price a gene therapy for which you have a one-time treatment? You might save a lot of money later, but somebody who produces this and needs to get engaged into the development of this drug has to spend much money, so how do they get reimbursed? So these type of things ultimately will have effects, and in my microenvironment in gene therapy, I find these effects already, because of you need a lot of upfront funding to, for example, make a clinical grade GMP viral vector. This is a very complex production. And this upfront funding is not necessarily there. It needs to be gained through, for example, MRC or Wellcome Trust, which has in a limited manner invested in these type of things. But then also early on engagement of commercial partners that will ultimately be required to make a drug out of an investigative product.
Good. We are actually moving on to the question of barriers to translation. So perhaps Professor Linden's given one answer. Perhaps I could ask the others to pick that up as well. What are the barriers to translation?
Well, if I just start, I mean, I think, so the great case that's quoted is monoclonal antibodies, and even then one might argue that development hasn't happened quickly enough. But and there, which I was involved with at an early stage, was the vision of the inventor who drove it. A basic scientist who discovered this technique by accident and ran with it and wanted to be involved in what we'd now call its translation or application, as really referred to. So certainly vision. So it's not just that there's lack of investment and money. I don't think that's the primary problem. I think if there's vision and that can inspire people, it's something real to be developed, money will follow because people will listen. So I wouldn't put that as the primary problem, but it's always a problem. He wasn't put off by the barriers that did exist, so we have a discontinuous system. Historically, we've got our educational system and universities, and we've got a health service, and they're separate, and there are means of bridging the two now through sort of bridging funding, which is absolutely fantastic, but it's still there's still a sort of cultural barrier, there's still a gap in actually moving something out from an inspirational beginning to something which is picked up and is shared as a vision by the people who will eventually use it. But that gap is closing. But that's certainly important. So we are stuck with a dual system, and probably it's too late to change it, but had we time all over again and could see what we see now, probably we would have a single system, a single employer set of rules with, at the one extreme, people working in universities on basic research, and the other extreme, providing health and not really involved in research. And I think that the point I'm trying to make, and probably we'll finish on, is context is very important. The fact now that basic science is going on, and for them to see the context, even if they don't want to push it themselves once they've got something really interesting, it's being in that context and knowing and finding someone to hand over the baton to, so that the race is completely finished. I think there are, more restrictions than just that to moving research on. It's knowing the right doors to tap on. One can spend years trying to find the right sort of support, and the right processes to help with, because all the processes and support, in my experience, can take years, and it's years and years and years and years on. And so I think that whole process of translation needs to be radically thought through to see if there are areas which can be speeded up. I don't just mean by funding, but just by process. So those are some of the issues. And I think, again, sorry, I will stop here, the organisational model is very, very important. Mm-hmm. So do we want something where we really believe in virtual networks across the country? Is that realistic? Or do we want geographical co-localisation that we can pop in and out of each offices and actually bring something together and inspire each other? So I think that model is very important.
Professor French-Constant.
Yes, so I would identify 5 barriers, and in fact this picks up on what's already been talked about. So first is partnership, and translation really does require partnership. Pharma and biotech need to partner with the academic community very strongly to provide the necessary bridge. And to be honest, I don't see that happening in the UK to the extent that I see it happening with my colleagues in Germany or in the US. And I think there are real issues there. Second, going back to Professor Sachs's point about organisation, I do think this is a major problem. The academic community has historically organised as a lot of quite small, quite self-sufficient groups. Very good for academic excellence, but much less appropriate for product development, which is what we're trying to do here. And I think there needs to really be quite high-level strategic leadership to persuade these groups to coalesce in a pathway. Thirdly, regulation. This is something that we've talked about before at another time. The regulations are very difficult, but I'd not only want to highlight the regulations around human cells, but also regulations about the use of animals. This is a major problem for us. Translation often requires being very fleet of foot in terms of the experiments that you want to do to get the major publications to interest the companies. And at present, if you have to do an experiment you haven't thought of in advance, which is obviously what happens in research, you need to get your animal license amended, and that can take a very long time indeed, and that's a significant delay in the process, and one that I think needs addressing really very urgently. Funding, I would agree. The problem with funding isn't the amount of funding, it's the way that it's being used, and I've already talked about that. And finally, people, particularly clinical academics. We talked about the gap closing between academics and the NHS, and the way that gap will close is by increasing the clinical academic community. And at the moment, it's still too small in this field.
Thank you. Dr. Bernay, is there anything you wish to add?
Yeah, I mean, yeah, I fully agree with what my colleagues have said. It's just that clearly there is a gap between what we can create in the lab and what needs to be done in the clinics. And the transfer of this prototype into a clinical product is the main challenge, especially with a really innovative medicine like embryonic stem cells or IPS, because basically we don't know what we need to do to reach the clinic. And that's really, I think, where is the problem, is that this access of knowledge and expertise and regulatory decisions that are very extremely complicated with very creative and innovative approach.
Thank you. Lord Winston wanted to come in.
Yes, I'd like to come back on a point that both Professor Sachs and Professor French-Constant have made, which I think is relevant. More and more universities, and particularly medical schools, have been exercised by the need for the research assessment exercise increasingly. So constantly a large number of clinical-based departments are being effectively run by non-clinicians, by scientists. And very often the clinical aspect of the work is, I think, in some ways relegated, and that vision is absent. Do you think that that has been a problem in trying to get clinical academics to run departments where you can see translation being thought about during the stages of the research?
Yes, I think that's is an important point, and just to be very specific about it, I think this business about health economic, well, I would call it prediction rather than assessment, is really important. In other words, health economic costing is usually associated with a fairly advanced clinical trial to see how much it saves and so on. But I think if one actually tries to go back to the preclinical work and to try to put some sort of scale on it and value, that will actually help to define some of the experiments and the developmental pathway it will follow. So I think that sort of looking back at trying to almost like a stock market, put a price on something before it's actually known what the price is important in terms of its interest in development, and probably just one example of the sort of thing you were talking about.
I'm going to turn to Lord Brose in a moment, but I think Lord Turnberg
Could I just...
Oh, sorry, did you
Sorry.
want to...
You asked the very specific point that you're getting clinical departments led by non-clinicians, and I think this is a real issue, and of course one of the major reasons why this happens is because the clinical workloads are so great for clinical academics that they simply can't do the very large administrative and leadership roles effectively, and the solution is basically to allow them the time, and if you do that, and actually in Edinburgh I think my bosses do this very well, then you can actually get clinicians leading basic science institutes, and I would argue doing so rather effectively and having the respect of both communities.
Thank you.
Yeah. Well, we are very pressed for time, I'm afraid, because I've got to get to the fourth question. Lord Turnberg had a brief point, then I'll turn to Lord Brose.
Professor French Constant, you set out very nicely the barriers. I wonder if you would like to offer some solutions.
That of course is a much harder question, and I think we need to devise mechanisms to persuade pharma and biotech to invest earlier in academic products, and I think one thing that they're looking for is a clear awareness in the academic community of what is and is not of potential value to them. So I think we have to both persuade the pharma and biotech to invest, and one way to do that would be targeted funding such as that the TSB provide, but equally we have to train our young academics to think much more about how you might form these links effectively.
Thank you. I'd like to turn to Lord Brose now.
The specific question that I'm asking is, can you give positive examples of interdisciplinary work? Is there sufficient collaboration between clinicians, scientists and engineers? And if not, how could this be encouraged? Now, we've really been discussing this to a certain extent, but I would make an observation. In my world of the physical sciences, and I'm a microelectronics engineer, transfer from research to practice was very often done by the people doing it. Scientists can be engineers. I heard a fascinating reminiscences about Andrew Huxley. I mean, he was an engineer in many ways. The same people can do things. Scientists can become practitioners. And, you know, I hear you saying, well, clinicians are clinicians. There's no way a clinician can really be a scientist, let alone an engineer. I would debate that issue. Would you like to comment? But the specific question, don't forget, is we'd like some specific examples.
Who would like to kick off? Maybe Professor Linden.
First, I'd like to get back to maybe answering your question and slightly giving a comment also to yours. So the development in biomedical medicines or products is actually quite complex, obviously, because we're dealing with a human product. So there are advanced therapies regulations, there are orphan drug regulations, very often the case. In many cases where diseases are being tried, there's pediatric regulations that are involved. And frankly, very often authorities and sponsors or investigators, we're both on a learning curve. And I think that's both sides are on a learning curve. And I think in many senses that's a good thing, because if the interaction is there and the discussion is there, one can find solutions to these. Yet the barriers go beyond just a single investigator, a single clinician, or a scientist. I think it addresses, at least in the field of gene therapy, many hurdles that come through regulations that have been set up not for this innovative type of medicines, but for longer term medicines, I think. And there need to be collaborations between authorities and investigators that can help move this forward. And the second point, many of the projects, at least that we're involved with, are quite international also. So you will have a gene therapy approach, and where a hospital in Sweden might, for example, be involved next to a hospital in the UK, which then means that there is a convergence between national frameworks that needs to be addressed in order to make such things possible.
Professor Sachs.
These are general points, but some examples may follow. One, I think, is alignment, because if you have a stream of money for forming medical MRC centres to push basic science towards translation, and two, at the same time, a very fortunate appearance of reorganised NIHR funds, if you can actually align them into a single stream, you complete the spectrum of activity, in theory. And in practice, I would suggest that it actually works, although it hasn't had sufficient time probably to see that. Secondly, that within that sort of framework, or even if it doesn't exist, I think it's important to have, well, people call them different things, but we call them workshops, other people call them clusters, to bring people from different disciplines together. So clinicians, basic scientists, computer, information technology experts, and so forth, students even, to bring them together, but focus not just for a bit of window shopping or window dressing together, but for a focus on a particular problem that they're all going to address. It might be to generate some money, it might be to unravel an experimental plan. And the more of this that is allowable, I think the more we'll see people working together. And it's very important that people from all disciplines have a buy-in, and they're not just going along to listen and to make an odd comment. They're part of the evolution of the problem at hand. And as I say, I think that there are some good examples at different centres where this is already going on.
Can you just mention one example of a sort of concrete success story in this sort of interdisciplinary
yes, so this is an example where in order to envisage an inflammatory system being activated and starting up, people who were involved in that inflammatory system, to be called complement, coupled up with people in imaging sciences who'd never really thought about transplantation before. And the object was simple, to try and devise a new imaging method with a camera held outside the body to see when this system lit up in tissues, when the inflammatory system became activated. And this was resulted with the help from cloning of molecules that can be used to identify this activation, to build a new system where this has become possible. It's still in animals, but it's easily, it's ready to be transferred to man. So, and there are other examples as well.
Thank you, Professor French-Constant.
So, I think where we're trying to actually repair a complex three-dimensional structure, I think interdisciplinarity does become particularly important, because we effectively need engineering approaches to either interrogate our therapy or indeed guide it. And I'd point particularly to substrate engineering and also to advances in imaging as being very successful examples of interdisciplinarity in this field. I think that's worked very well. I think there is an enormous need for much more of it, but it seems to me that this is something where the community has appreciated the importance of working with chemists, physicists and bioengineers. and is doing so quite effectively. And there's an outstanding bioengineering group at Imperial would be an example, who are doing fantastic work in this area. Going back to the specific point that was raised about, well, why don't the scientists get out there and do the business? I completely agree with you. My observation, though, would be that those who try and do that have to spend so long on the process of trying to get their business off the ground and keep it alive that their science suffers very badly. And as an institute, as a centre director, this is something I worry about a lot. I see very bright young scientists that I have in the centre trying to start companies and spending so much time having to work on that company going that it definitely affects their primary scientific output.
Dr. Vallier.
So in terms of example of multidisciplinary activity, in Cambridge we have a strong interconnection between clinician and biotech, and our recent work now have shown that we can work together to show that we can use, for example, hepatocyte for personalized medicine using IPS. So at least what works very well is training of clinicians and during their PhD in a stem cell and academic lab, and that's developed a very strong interconnection and strong knowledge. I think the key problem then is that often these clinicians go back to the clinic, and the key problem is that being a basic scientist and a clinician is two full-time jobs. And to be leader in one of these jobs means that you have to spend all your energy on one specific job. And that's the key problem. And we see this discontinuity. So in the stem cell field, it's such a competitive and aggressive field that you can't just step back from your research for 2 years, go back to the clinic and hope to be competitive and be leader in science at this time. And that's one of the key issues, how you combine your academic life with your clinical life. Because you can be the translational aspect won't be solved by a clinician that become full-time basic scientists, because they will lose their connection with the clinic. They have to remain clinician at the same time. And that's one of the key challenges I think we are facing at the moment.
Could I come back on that, Chairman? This is an issue of sort of career development, isn't it? Professor French Constant, you said, the scientist. Well, you can be a scientist for 15 years, and then you can go and do something else, And in many fields, it has been the outstanding scientists who've made their contribution. They don't necessarily have to come back again, and life goes on a long time. You can do a lot of things in your life. But you, in the medical field, to be a bit contentious, to put yourselves very much in boxes,
That's because we're continuously learning, I think.
Well, that's the same, exactly what happens if you look what happened in my field, how, we went from one transistor to putting 256,000 million of them on one chip. There was a huge amount of science, a huge amount of development, but that was done very often by the very clever people who went on and did that.
Could I ask just a brief reply, Professor French? How do you top that? Yes.
I think, yes, if one had been in science for 15 years and was clearly very established, then that's fine. But when you're 2 or 3 years out, just starting your own independent career, and you need your next fellowship in order for the university to give you any form of long-term support, I think spending half your time trying to set up a company is a very high-risk strategy indeed.
Lord Dixon-Smith.
Chair, this may be a somewhat tangential question, but I haven't heard whether there is, or there may well be, constant liaison with your regulators. It seems, I do understand the conflict of interest to a certain extent between what I would call pure academia and the financial side of life. But if you haven't actually got the regulators on side and aware of any potential problems long before they become real problems, you've got a very sticky wicket to bat on with both the other sides.
Anybody wish to offer an comment to that?
I would observe that there's actually a meeting going on today where the community and the regulators are talking. So there is certainly a clear understanding of the problems here. Where you have a regulatory map that looks like a rather complex underground system, it is actually very difficult to navigate your way through it, and I think simplicity is required in order to make these interactions easier.
final question from Lord Willis.
Just a very brief one to Professor Sachs. All speakers this morning all witnesses this morning have emphasised just how silo-based so much of our approach to these areas are, both the difference between clinicians and indeed academics, also the difference in terms of funding bodies. You mentioned, I don't know whether it was a slip or I misheard it, that one of the ways would in fact be to pull together both NIHR funding and MRC funding in order to create, as indeed was Cooksey suggested, a single body. Was that a serious suggestion, and did the rest of the panel support it?
Well, it was a serious suggestion, and it actually has happened on a number of sites where the two do exist. And I think it works very well. And it's a logical development, because if you have one packet of money for developing a more basic concept and another packet to bring ideas into practice, then what better way than to co-localise them? It's a very efficient way of spending public money, as far as I can see.
Is that shared?
I will have some concern in terms, how you maintain the balance between basic research and clinical research, because the NIHR have a very clear objective, and the MRCs have a much broader in terms of application and development. And I would be concerned that this aspect disappear if we only focus on a fused NIHR MRC unit.
I wasn't proposing this as a total solution, just as a part of each package, which exists anyway. Yes.
I think we've actually run out of time, so I would like to draw this session to a close and thank our four witnesses very much indeed for their answers. If there are any points that you feel you haven't been given time to express, please do feel free to write in and add to your evidence, which would be very helpful to us. And in due course, you will receive a draft of the transcript for any corrections you wish to make. So thank you very much indeed, and I'll allow you to leave the room before we invite on the second witness panel. So thank you.
Thank you.
I'd like to welcome our second witness panel and to repeat what I said at the beginning of the previous session. Members of the Select Committee are urged to declare any relevant interest before they speak for the first time in this inquiry. This is an inquiry into the topic of regenerative medicine, and the focus of this session is going to be around the public understanding, in part, of what is available through regenerative medicine. So we're going to ask you about certain articles that appeared in the press and trying to understand what actually is the reality, as opposed to the hype in presenting to the public what's going on in this field. What I'd like to do is to invite you to introduce yourselves for the record, and if you wish to say anything in addition to introducing yourselves, please feel free to do so, but keep your comments brief, because we've got a limited amount of time. And I'd also like to just remind you that in the second part of the session, say for the last 20 minutes or so, we have a number of questions that were submitted by the public through Twitter, and we want to put some of those questions to you so that those who have submitted the questions, who I hope will be watching the webcast of this session, will have a chance to see their questions being put before top experts and hearing your responses to them. We won't be able to cover all the questions because of time, but we will cover a number of them. So without further ado, could I ask you to introduce yourself, starting with Professor Kaur.
I'm Professor Peng-T Kaur. I'm a clinician scientist working in an area called glaucoma, but I have an interest in regenerative medicine and also drug therapeutics to enable that and prevent scarring. I also direct the NIHR Biomedical Research Center in Ophthalmology at Moorfields Eye Hospital and the UCL Institute, which has the express purpose of rapid tracking, translation, and hence the infrastructure behind that. Thank you.
Thank you. Professor Barker.
I'm Professor Roger Barker. I'm a Professor of Clinical Neuroscience at Cambridge University. I'm a clinician, a neurologist who sees patients, and for the last 20 years I've been involved with regenerative medicine around Parkinson's and Huntington's disease, and I coordinate the European consortium on a fetal transplant trial in Parkinson's disease.
I'm Michael Schneider, the British Heart Foundation Chair in Regenerative Cardiology at Imperial, and as a member of MRC Council, I was a participant in the Research Council's Technology Strategy Board working group that crafted the UK strategy in regenerative medicine that was alluded to in the earlier session.
Good, thank you very much. Well, perhaps I could kick off with the first question, which relates to the press articles that were sent to you. There were 3 articles, one from the Guardian, one from the Daily Mail, and one from the BBC. And what we'd like to ask you is, could you comment on the treatments discussed in each of the articles, and particularly when they might be available to patients on the NHS, and whether the articles portray a realistic view of the efficacy of the treatments. So the Guardian one talked about improved vision for a man with Stargardt's disease, and the Mail one talked about Parkinson's disease, and the BBC one talked about heart attack scars. So perhaps on the question of treating diseases of the eye, I could turn to Professor Kaur to talk about the Guardian report by Sarah Bosley.
I think Sarah Bosley's article was a good one. I think it reflected some of the clinical issues facing us and our patients' desires. I think it reflects very well on how passionate our patients feel about wanting stem cells. There isn't a week that goes past that I don't get asked. And it also reflected quite nicely on some of the difficulties facing translation of stem cell research. I think going on this particular issue, I mean, going back to the patient, to the public's desire, we meet the public, the board of Moorfields meet the public every year, and an elderly gentleman got up and said, I have age-related macular degeneration, is there any hope for me? Is there going to be stem cell treatment in the public forum? And I answered that, but I said, unusually, I'm not going to tell you 5 to 10 years' time. I'm going to tell you that within 18 months, we're going to have the first clinical trial. I was wrong, because actually we started the first clinical trial 6 months after I made that statement. Now, that's a very unusual situation for us here, but it is a reflection of the rapidity at which, at least certainly within the eye fraternity, regenerative medicines, gene therapy, we did the world's first patient a few years ago, is moving forward and moving forward rapidly. Going back to the articles, I think the public is inevitably very, very hopeful about this. And even though the early reports of this trial by ACT, which was, they reported the first 2 patients in the Lancet, which was done by a former alumnus of us in Los Angeles. And then the ACT people came across to our biomedical center, because obviously we're very big and we have a lot of expertise in this area to carry out the first trial in Europe. The early reports have been, these trials are obviously for safety. They're not for therapy, but inevitably in these very frontline treatments, and I include gene therapy in that, we begin to see some very interesting results. Now that naturally raises the hopes of the public a lot, because it in many ways, a safety trial also accelerates expectation, because some of the patients have experienced quite interesting improvements in their vision. So they've been very helpful in some ways, because they've shown that proof in principle it's possible to, going back to the stem cell therapy, it's possible to do it. Secondarily, they've not been overtly rejected, which is obviously a major concern. Thirdly, at the moment, there don't appear to be any tumors or any of the other issues that we've seen so far. So that's very helpful. But obviously, there are still significant challenges moving forward to the future. And of course, the issues of whether we can really get this through to full clinical use within the NHS. I think that will depend on the whole licensing issue, other issues of funding for both researchers and government. But I mean, there's no question that the desire is there. I mean, that gentleman's question reflects the desires of, I'll say literally millions of people around the world for this treatment to work. Mm-hmm. And therefore, I suppose it's within our scope here to try and see how we can make that a reality. But I think it's not just hype. I think, going back to your initial question, there's real hope. We are proceeding. We're right at the front line. There's a lot more to do, but and I think some of the problems have been identified here, but certainly we're moving fast and moving forward.
So in terms of a specific answer to the question of when might the treatments be available to patients on the NHS, you've said you hope that in the future they would be, yes, but you haven't sort of hazarded whether that's 5, 10, or how many years off.
Well, I think the trials are moving quickly, and I think one of the interesting things about stem cell therapy is that, unlike pharmaceuticals, you probably, because you're going for regeneration, you need perhaps less patients. It's still very, very difficult to do these trials because you need very sophisticated analysis techniques to look at efficacy. But you don't need so many patients. Now, this is important back to your question, Chairman, because I think if the safety trial goes all right, and that these will complete, hopefully in perhaps in the next year or so, then hopefully the companies will be moving forward to the later phase trials. And clearly, if they do show efficacy, and that depends on regulatory issues both here and around the world, then potentially the treatment will show efficacy if they succeed, and then they'll go through the normal licensing. And I think, given the speed at which you can go, given the small number of patients, it is possible that treatments like this will be available within the next 5 years, at least licensed. I mean, there are other issues, of course, governing availability of treatments, such as cost, etc. But I think there is a distinct possibility that they could be available within the next five years. Mm-hmm. And I think that's an exciting possibility for all of us.
Okay, thank you. Now I'd like to move on to the second article and ask Professor Barker to comment. This is the Mail article by Sadie Whiteblocks about hope of Parkinson's treatment breakthrough as human stem cells successfully used to treat the disease.
Yeah, so I mean, this refers to this very influential and important paper that Lawrence Studer's group published last year in Nature. And as often the case with newspaper articles, the headline is somewhat distorting exactly, I think, what the article was saying. So in this field of Parkinson's disease, the use of cell-based therapies to repair the damage in Parkinson's has been going on for 25 years. So in Parkinson's disease, there's a specific loss of a cell that produces a specific chemical, so any cell that could produce that chemical, in theory, could repair the brain. So for 15 years up to the turn of this century, fetal dopamine cells were being used with mixed efficacy, and many people have got confused with the idea that fetal tissue is the same as stem cell-based therapies. So in the public's mind, there is a confusion between, when you talk about a fetal-based cell-based therapy and a stem cell therapy, are the two one of the same, and haven't stem cells been around for a long time with Parkinson's disease? The big problem in the field of Parkinson's disease has been twofold, I would say. One is, do fetal dopamine cells actually work in Parkinson's disease? And I would say the answer is yes, but it's not consistent, and that is the reason for our trial. And secondly, in the field of stem cell therapies, are those stem cells which you are producing truly of the right character to produce the same efficacy as you might see with a fetal dopamine cell? So the problem with stem cells is obviously to do with tumours and things of that nature. In Parkinson's disease, the critical question has been, have you been able to make a dopamine cell which is of the right type to treat Parkinson's disease? Not just any dopamine cell, but an appropriate dopamine cell. And this paper by Lawrence Studer took a new approach, which was to take a developmental biology approach and say, how do dopamine cells normally develop? Let's recapitulate that. And they did that successfully using human embryonic stem cells, transplanted them into immunodeficient rat mice and immunosuppressed monkeys for a short period of time, and showed that the transplant survived, made dopamine, and restored deficits in those animal models, which are a crude approximation of what you see in the clinic. So this paper was important because it demonstrated that, and this paper makes that quite clear. The problem then is, well, when will that translate to the clinic? And I think there are a couple of issues to say there. One is we don't know whether dopamine cell therapies truly work, and that's the purpose of our trial. Secondly, the stem cells, the dopamine cells as produced from these stem cells, still do not have all the hallmarks of normal dopamine cells, so there are still issues to do with that. So when will these treatments come into the clinic? It is envisaged that there will be early trials using this technology, I imagine, in patients at some point in the next 5 to 10 years. But they will be trials. Lawrence Studer himself has applied for funding to do that through the New York Stem Cell Initiative. We have just got a grant from the MRC to take Roslin cells, GMP grade cells, human embryonic stem cells, through to dopamine cells to transplant into animal models. So we have a cell that is of clinical grade which could go to the clinic. If that all goes according to plan and those cells work, then I think the fundamental question which will face the NHS is that treatment any better than any other dopamine therapy that's out there? So I think there is clearly in my mind an issue of you can get something to work, but then it will be, is it competitive? Yeah. So for Parkinson's disease, as you may know, whilst the loss of dopamine can be remedied with this type of approaches, the disease is much more diffuse than that. The dementia is quite common in Parkinson's disease, and that is independent of that. So all therapies for Parkinson's disease which are currently available in the clinic work on the principle of replacing dopamine. And so fundamentally, will a stem cell-based therapy be any better than a duodopa infusion, an apomorphine pump, better oral therapies for dopamine?
Okay, thank you very much. And Professor Schneider, would you like to comment on the BBC News article, Stem Cells Used to Heal Heart Attack Scars, healing quotes?
Yes, the trial being discussed is the Caduceus trial, led by Eduardo Marban in UCLA. And to put its importance into perspective, I would say over the past decade, there have been more than 1,000 patients treated worldwide with stem cells of different kinds for heart repair, typically bone marrow cells or their circulating derivatives in the bloodstream. The consensus from those trials is that small benefits are seen and that they overall are safe, but the benefits might be less than enthusiasts would have wished, partly because the bone marrow cells probably don't turn into beating cardiac muscle cells and therefore don't achieve myocyte replacement as might be necessary for the optimal restitution of heart function after a heart attack. This particular trial was one of 2 reported in the Lancet this past year that used instead dormant stem cells from adult heart muscle along with other cells grown from a cardiac biopsy and have very well proven potential to form new beating heart muscle, both in tissue culture experiments and in experimental animals. And the point to which the title of the BBC report made reference was the shrinking of the scar by advanced magnetic resonance imaging technologies. So I agree very much with the caveats that Professor Kaw raised. These are phase 1 trials that are designed and powered chiefly to prove safety and gain the approval to go ahead from a few dozen patients to a few hundred patients in the phase 2 study, and it's really only in the phase 3 study involving thousands and sometimes tens of thousands of patients that we get genuine proof of clinical effectiveness. Piggybacked on the safety trial, however, are studies designed to provide a preliminary indication of effectiveness, such as the imaging that was done in the case of this trial. And the problem is partly that the media don't always make it clear to the public what a trial is and isn't. A phase 1 safety trial is not proof of effectiveness. It can contain a preliminary indication of effectiveness, but those aren't the same thing.
Mm-hmm. Thank you very much. A brief question from Lord Winston, then I'd like to turn to Baroness Sharp.
Two brief questions, really, my Lord Chairman, if I might. One is
questions brief and answers brief, please.
One is the paper by Kim, published over 10 years ago in Nature, on tyrosine hydroxylase activity in dopamine. We remember this paper, which showed clear efficacy in the rat for treatment of Parkinson's disease. One of the issues is that actually it's over 10 years ago. It takes a hell of a long time, and I wonder if you'd address that. And to Michael Snyder very briefly, one of the issues, as we've discussed in the past together, is the fact that the early trials with stem cells, whatever kind of stem cells injected, all looked rather good, and there was all sorts of parameters which showed an improvement. But actually, once randomized controlled trials were done, actually the improvements, if at all, were extremely small.
Okay, Professor Barker.
Yes, I mean, there are always reports on there. I mean, this paper was from Ron Mackay's lab, and the ability to turn a stem cell into a Th positive, which is the enzyme which makes dopamine cell, has been shown many times, and they do have efficacy in these animal models. And in order to reverse the behaviour in an animal, you only need 160 Th positive cells to effect that. In order to have a successful transplant in a patient based on the fetal material, you need 100,000 Th positive cells. So extrapolating from animal models to patients is always very difficult. It's a starting point, but it's the reproducibility of that data, of that technique, protocol in other people's hands. And in those particular cells, really what the cells look like was not the same as what Lawrence Studer's managed to do with his cells. But his cells still do not look like normal nigral dopamine cells.
Professor Schneider on the other.
So turning to the small effect of bone marrow in the early clinical trials for heart repair, I would agree with your point that even preliminary indications of efficacy are more convincing if randomized rather than not. However, I would say that a lot is learned even from the early trials that affect the design of later trials. So for instance, although the overall effect of bone marrow was just a small improvement in the ability of the heart to eject blood each time it contracts, other kinds of improvements can be detected more readily in the trials, such as protecting the heart from progressive dilatation and wall thinning. That effect is a big one, not a small one. And the early trials also helped pinpoint those patients who might be most likely to have a big improvement. So if you inject bone marrow cells in someone who has a small, uncomplicated heart attack, whose heart is pumping well already, there's not much benefit. And those patients, in turn, diluted out the benefit that's seen in patients who have a big heart attack, whose hearts are pumping worst, in whom the benefit was largest. So in talking about the early bone marrow trials, I think a lot was learned that affects the design of the trials we see today.
Baroness Shaw.
Yes. Well, these sort of articles are leading to a lot of interest, obviously, in stem cell therapies of one sort or another. And the question I want to put to you is whether there are dangers for the public travelling overseas. There are quite a number of places overseas that have been setting up clinics of one sort or another, and we've seen this happening. And are there dangers for the public to travel overseas to have regenerative treatments of this sort? And to some extent, no. If so, what can be done about this?
Well, I think you're absolutely right. I think there's a danger to the patients. I think there's a danger to the whole field. Because if something goes wrong in one of these clinics, it will impinge on everyone who's working on stem cell therapies across the globe, regardless of the origin of that transplant. This is a very big problem, and I've been involved in it in two levels. I've been to China twice, where there are a lot of these clinics, to talk to the clinicians out there to see if I can persuade them to try and adopt a slightly different strategy without success. And the other way in which I have done this is through the International Stem Cell organisations have tried to set up guidelines to help patients decide whether something advertised on the web is worth pursuing. And it's very difficult because I have one patient, for example, who has young onset motor neurone disease, and she came to see me and said, I want to go and have an umbilical cord stem cell transplant. Would you support that? And I would say, no, there is absolutely no scientific evidence that that works at all. She said, is there any possibility it could work? And you said, well, of course there is a possibility it could work. And she said, and you're offering me nothing. So some possibility against no possibility seems quite good odds to a lot of people, even if it costs £20,000–25,000. So I think this is very difficult because within those clinics there may be something useful coming out which we cannot detect. The way in which the trials are done, certainly in the clinics I've been to, has no scientific or clinical basis. The follow-up is minimal, and the results, I think, have many interpretations, not the interpretation that they necessarily put upon it. So I think it sets up an expectation, and linked into that with the patients are two issues which I've picked up on. One is that, first of all, you don't get it in Britain because it's of the NHS doesn't have any money. That's why I'm having to go abroad to get it. So it's the cash-strapped NHS, which means I have to go abroad. If we had enough money, we would all have it, and you have to disabuse them of that idea. And secondly, it's to do with the ethics, that we're so wrapped up in the ethics of human embryonic stem cells, fetal tissue. Actually, these adult stem cells, bone marrow, whatever, which are being offered in other parts of the world, circumvent that problem, and therefore they must be a better source of cells to use. So the ethical, and that's what the clinics often play on. They play on this ethical issue to get around it, to say these are much more ethically acceptable. There's no scientific basis, but they're ethically more acceptable, have them. So it's a big problem, and this stem cell tourism, people travelling the globe to see it, and it is vast. I mean, when I was on this committee, I said, send me the clinics. It was 64 pages of listed clinics across the globe. So this is a vast industry that's out there.
Would either of the other two members of the panel like to offer additional comments on it?
Well, I mean, certainly in eye disease and certainly optic nerve disease, which currently is irreversible, obviously we do have many patients writing constantly and then saying, should I go, should I go? And I think I'll reiterate a lot of I won't go over the comments that Professor Barker has been through, but I mean, I would absolutely reiterate that issue. It's a difficult situation for us when you have a relatively an untreatable condition and a patient is desperate. I suppose my reiteration for that would be that actually the way to try and go through this is the issue of information. Is to provide the individual as much information as possible about what we know about where they're going. That includes the individuals running it, and also what they know what's available out there. And I think that's the first thing that's been very helpful, perhaps in informing people. The second thing is the personal approach, actually, because I mean, when they're in very desperate state, people, well, they want to ask you, if it's your mother or your child, would you send them? And actually, the answer, I look at things and I explain to them the information. I said, although I'm not in your position, my answer is I would not, and this is why. And that's actually as persuasive, to be honest, as I give them the information, but it's that very personal approach that's helpful. So I think they need information. They need recourse to clinicians who are familiar with the area, familiar with the science, and actually can give them a very frank personal opinion also about the pros and cons of this.
Professor Schneider.
I share the concern that Professor Barker raised that safety accidents in this field in unregulated clinics with poorly designed trials could have an adverse effect, and I suppose to an extent we're fortunate that there haven't been more than have been reported to date. There is one instance that I'm aware of a young man who went to the former Soviet Union for injections of fetal neural stem cells for a neurological disorder, subsequently developed a tumor which advanced pathological tests were capable of showing were donor cell derived. And if one thinks back on the history of gene therapy and the recoiling of the investigative community and the clinical community from gene therapy based on a very small number of safety disasters, the same could happen here. And so these kinds of activities do put an entire promising field in some jeopardy.
Yeah. Okay, thank you. May I then move on to Lord Selborne?
The Guardian article refers to a tide of suspicion to stem cell therapy and the need for practitioners to fight back. Is the tide on the ebb or the flow at the moment? And what research has been done on public perceptions and attitudes to regenerative treatments? What does that tell us?
Well, I have limited experience of this. As part of our European fetal transplant trial, we have an ethical work package within it, and the EU has been quite keen on collecting public opinion on regenerative medicine. So my colleague, Professor Gottweiss, who's a professor of life sciences in Vienna, pointed me in the direction of a thing called the EU Barometer, which looked at various different aspects of this, which I'm sure many people are familiar with. And certainly within the UK, the support for stem cell research and stem cell therapies is very prominent. I mean, you know, it's the highest in Europe, and this barometer, 80% of people would support the use of stem cells for research and for therapies. I think what would be interesting is when you actually move to the clinic, how many people would support it on an individual basis. So, for example, with the fetal transplant trial, whilst we have lots of people who've joined our trial, when you actually say, would you like a fetal transplant, because we're about to start, people just step back a little bit from that. So I think people are generally very, certainly in the world of neurology and in general with stem cell therapies, people are very supportive of it. I think when it comes down to an individual person in a trial, then I think it changes slightly. But in general, I think people are supportive of it, but it does vary across Europe, and with a lot of funding for European-wide trials, this creates a certain tension in how you move some of this work forward.
Would further public engagement be helpful to meet some of these issues that you're referring to when it gets to clinical stage?
Well, I'm a huge fan of public engagement with it because I think there's a lot of misconceptions about things. There's a lot of things we don't think about as scientists and clinicians, which the public are very good at educating us on thinking differently about it. I think managing expectations is the most important thing with these type of therapies because, as the papers tend to say, and I always have, when I give a talk, there's a headline from The Guardian which says, miraculous cure turns into disaster, which was a fetal transplant trial in America. And as I say, well, there are essentially two outcomes often in the papers. It's either miraculous or disastrous. And therefore the public have this hyperbole. It's either going to cure everything or cure nothing. And so trying to give them some sort of realistic expectation of what it could do over what period of time, in what aspect of the disease, I think is important.
Going back, we, as part of the NIHL, we have several very large public engagement days on different diseases, inflammation, retinal disease. Now, obviously, these groups of patients have a particular interest because they suffer from these. So you might expect them to be more engaged about wanting the treatments. But there's often a very active debate about, should we be wanting stem cell treatment? But I can tell you that at the end of the day, we often have a session where we ask them to take their post-it and stick it on the bits of the board that they are really keen on. And of course, research comes very high, interestingly, even though they're all sufferers of the disease. And I think for the few sort of informal surveys we've done on them, certainly they're all very in favor of stem cell therapy. In ophthalmology itself, we haven't done a large-scale public analysis of whether this is is the same sentiments are held. But I mean, again, I can tell you personally, and certainly through our wide exposure to patients, through our public patient engagement, there's very strong support for this because they do see it as giving them some hope, particularly in many of the conditions which are essentially untreatable. So there's very strong support, certainly from the patient groups.
And Professor Schneider, do you wish to add?
Yeah, I would share your point that there is insufficient public knowledge of the nuances of clinical research of many kinds, and this is something which most of us in academic medicine are resourced to do either through our NHS trusts or the NIH biomedical research centers and biomedical research units. I think back to Lord Winston's program of 4 or 5 years ago, Super Doctors, which painstakingly made the point that in a clinical trial, half the patients don't get the treatment, and that indeed would have been a surprise to many members of the public. So there are many things that the public would benefit from understanding better, and we as a community would benefit from improving their knowledge.
Lord Patel.
Because the question was put as in public attitude to regenerative medicine, it implied we're talking mostly about treatment, but we're not, of course. We're talking about research too. So in the context of research, what engagement do research funding bodies, including the councils, engage with the public before the research starts? Are you aware of that?
Yeah, I don't know a specific answer to that. I mean, to be honest, I have more to do with the disease-specific charities. So things like Parkinson's UK, for example, I often go and give public lectures to people with Parkinson's disease and interested parties about regenerative medicine and things of that nature. I don't know so much about the MRC and the Wellcome Trust, whether they have specific public outreach sort of symposia or days, I'm afraid.
Any other comment from other panel members?
Well, I mean, I think true that, again, I'm not so sure about Wellcome and MLC, but certainly through the NIHR, there are very proactive PPI engagement activities through modern, web media and other things. And we've got priority setting partnerships, for instance, which is the public has been engaging very, very strongly in by emailing in what they feel really matters to them in their lives in terms of research. And that priority setting process is underway at the moment and will identify some key priorities which hopefully many of which will be funded by the NIHR and other sources.
I think in a small number of areas the ethical issues loom large, and so that kind of consultation takes place in a proactive way. The recent Academy of Medical Sciences report on animal-human hybrids would be one example. Fetal tissue research is another example, and of course human embryonic stem cells would be a third. But as several speakers have pointed out, this field of work, more so than others, is very, very dependent on high-quality animal research, both for model organisms, providing us cell types of different kinds which one can study and then learn from the human, model organisms like the salamander, the zebrafish, and even the newborn mouse, which have a much greater regenerative capacity than middle-aged or aged humans. And so we learn from the model organisms mechanisms of repair that one might then try to apply to the clinic. And in addition to those two streams of fundamental research requiring the animal models, I would point to the need for large animal studies to develop the technologies for delivering cells safely and prudently and effectively to diseased sites in diseased organs, so that the kinds of treatments that we're talking about virtually couldn't exist without three strong streams of animal-based research leading to the first in human innovations. And that's an area where I think increased public awareness of these needs might help to shape public opinion in the future as more tolerant of the need for animal studies.
Okay, I'd like now to move to the Twitter questions that we received, and I think you've had copies of. We are very pleased that we had a good response to this experimental request for input on Twitter. We probably won't get through all the questions in the remaining 20 minutes or so, but we'll pick out some to start with and see how we get on. And I hope you'll be able to give us reasonably succinct answers. Some of them reflect points that we have covered already, but it would be good to recap them in the context of these questions. So I'm going to turn to Lord Rees to put the first one from our list.
Yes, here is one. Is there a regenerative medicine research program with measurable and realistic goals? Should research be focused on one condition to concentrate efforts?
So I'd like to respond to the second of those first. I think in regenerative medicine, the mechanisms of benefit vary from cell replacement to indirect effects on the host tissue, including blood vessel formation and wound healing. So I would argue against picking one prototype disease as allowing us to solve all the problems in the area. Turning to the questioner's first question, is there a good roadmap working forward? I think the UK strategy for regenerative medicine, which includes not only injecting a naked suspension of cells, as it were, into the diseased tissue, but also cell-free therapies based on the hormone-like proteins that stem cells make, and also engineered constructs having more of the three-dimensional configuration of the tissue. I think that's a very comprehensive plan that incorporates many of the elements of regenerative medicine, and it is a very good roadmap if adequately resourced.
Professor Parker.
Yeah, I mean, I would support those views entirely. I think concentrating on a few conditions within one area of medicine. So in the world of neurology, Parkinson's disease is an obvious target because it has a specific pathology. So I think targeting one condition in a certain area is a sensible thing to do. I think we have a very useful way of taking it forward. I think one of the key challenges of the future is how you measure whether your therapy has really worked in the time frame that we think about. So if you put a cell into someone's brain to repair the brain, if I give you a tablet for your high blood pressure, you would expect to see a result within a few weeks. If I put a cell in your brain to make it repair that bit of the brain, it may take 5, 10, 15 years before you see the benefits, true benefits. So thinking how we can actually do trials better, I think, is a real challenge. And that, I think, has been a problem in the American system, where trials have been ended prematurely based on how they see drugs working, which has had major consequences on regenerative medicine trials across the planet.
Professor Kaur, is there anything you'd add to this?
Again, I'll just start with the second question first, just like Professor Schneider. I think that it's a mistake to say that just because the disease area is currently costing you a lot in the area. I'm not to say that's not an important factor, that's very important. But I think the issue of rare diseases is a very important one. The UK has a unique ability to see and bring together groups of rare diseases. And I'll give you an example. The world's first gene therapy in a human done done in the UK was a very rare condition. It's only because we are able to gather in the UK these very rare conditions together, we're able to do these early proof of principle. Rare diseases are often an incredibly good way of finding a proof in principle of a new treatment, because they're often very dramatic, they're very precise, and then once you have that proof in principle, you can apply that principle to much, much more common diseases. So some of the things we've learned in gene therapy of these incredibly rare diseases could now be applicable, for instance, in potentially the treatment of the retinopathy of diabetes, which is a major blinding problem in young adults in this country. So I wouldn't stress just on the diseases because it's very common, and it's very, because there are paradigms of disease, particularly in the UK, and the ways the health system is set up that could make us be able to compete very effectively. The second question, which is back to the first, is that are there programs with measurable and realistic goals? I think the answer for all of us is absolutely yes. The trials you've heard about, the London project, many of the other stem cells have very clear goals for progressing and translating through to clinical use. And perhaps we'll touch on that. But I mean, the issue about, I would like to just raise one point about that, and that translational research, which you raised today in your committee, is not an accident. It's something you do because you want to do it so much, it burns in your heart and it hurts that you want it so much. You know, I suppose the analogy of translational research is it's a bridge of which every single rung has to be in position. That makes it different from some of the really world-beating, science that you've seen. Insofar as, I'm not to denigrate science, science is extraordinary, but to make things translate, if one rung of your bridge is missing, you will fail. And so it has to be very, very deliberate, it has to be recognized, it has to be cultural, and often the people who put those rungs into place aren't recognized or aren't provided. And I think to make translational research work, you just have to want to do it so much that you have to put all the rungs in position so that it works. And that's how we've succeeded in some of the things we've been doing.
Okay, I'd like to turn to the next question and ask Lord Turnbow to pose that.
Thank you. This question, I think, goes to Professor Kaur. And I think we may have covered some of it when in your first remarks.
Yeah.
And it's about the FDA granting orphan status for human embryonic stem cells for treatment of inherited macular degeneration in children, Stargardt's disease, And are we going to see this used in the UK for compassionate grounds?
Well, I mean, clearly if you're a sufferer of Stargardt's, you're reading at least good safety reports so far, albeit reasonably small numbers, and you're thinking, can I have this treatment now, please? Which is what our patients ask us. Obviously, for a drug to be made available for compassionate use, many things have to be placed. First and foremost, of course, you have to have enough data to, on the balance, be sure that it's safe, first and foremost, and that there's at least some evidence of efficacy. We're moving towards that, but obviously that takes time. And so I suppose the answer to that question is further analysis and further completion of the trials, the current trials, and perhaps one further are needed before companies can provide this treatment for compassionate use. But obviously it's a possibility once we reach the state of evidence where that might be possible.
What is Stargardt's disease?
Stargardt's disease is basically a form of macular degeneration that occurs in young people. And so it's a good model insofar as, well, it's not a good one. I mean, obviously it's very tragic and it's very devastating for young people who lose their central vision, the vision we're all using now to read our papers and do everything. So, but it's a very specific condition. It's relatively rare, but it's obviously a very good condition in which to do early phase stem cell trial.
And does the fact that the FDA has granted this orphan status, does that mean they've assessed the efficacy and safety?
No, I think orphan status purely refers to a disease that is sufficiently rare.
Yes.
I think going behind the thinking behind both the European and the FDA definition of orphan is that nobody would develop any treatments for rare diseases if they didn't make the pathway a little bit easier. Yeah. So the route, if you like, to licensing on an orphan disease is a little bit easier. And I think that's the main issue about the orphan disease.
Lord Winston.
My Lord Chairman, I've got Twitter running in front of me, which is why I'm asking the question, and I see that one of the, at least more than one, people quote Professor Yamanaka from Japan.
Sure.
I hope one day is to use iPS cells rather than human embryonic stem cells. I wonder if you'd like to comment on that specifically, because it relates to this particular respondent as well.
I mean, obviously we were very thrilled by the award of the Nobel Prize to Shinya Yamanaka for his very revolutionary work on iPS cells. iPS cells obviously have raised a lot of hope because of the fact that you can take adult cells and convert them into multipotent cells that can do various things. I think there are a lot of technical issues, some of which are probably beyond me, about the iPS cells, but there are a lot of technical hurdles with iPS cells at the moment. It isn't just because, embryonic cells are just because they're available, but at the moment we know a lot more about the current cells that we're using, which are, take for the ACT trial in the London project, which are embryonic derived, and therefore we're much more able to move forward relatively safely. I think with the iPS cell technology, we're still very uncertain about how to go. There's a lot of work obviously being done on iPS cells and its potential for therapy, but I think at the moment we're not in a position to be able to move forward with iPS cells therapeutically.
Okay, I'd like to turn to Baroness Perry.
You touched on several times in your replies. I think, Professor Barker, you talked about the importance of managing people's expectations and so on. The question is there a need for improved patient education on the whole subject of regenerative medicine? Should there be better science communication strategies designed to provide accurate and realistic messages about such technologies to the public?
Yeah, I mean, I think it's a very dynamic process because this field moves forward. 5 years ago, no one knew what an iPS cell was. The term did not exist. Well, no, that's not true. 2006, but a few years ago. So people understanding what these terms mean and what they bring to the field, I think, is very important. And I think education is very important, especially as we move in a world or live in a world where there's a lot of commercialisation. So the other thing which happens with stem cells is they obviously, some of them get attached to companies, and once they get attached to companies, there's generally only one piece of news you'll hear about those stem cells, which is good news. And so again, it is this building up of expectations that there are lots of things out there, but if only I had access to it, I would be much better treated. So it's great when you have people, my colleagues here on the panel, who patients can talk to and say, what do you think of that? But having a broader remit whereby one can educate people and understand what they mean by stem cells. So an embryonic stem cell is different from an induced pluripotent stem cell, is different from a neural stem cell, is different from a bone marrow mesenchymal stem cell. These are all different stem cells. They all have the same term and people group them together. And so I think educating people on the science, what is a stem cell, what can you do with it, educating people on where you're trying to take that therapy, that cell into the clinic, and then the realistic expectation of what that would actually mean to a patient in the short term and in the long term, and the risks that that brings with it.
Professor Schneider, do you want to add anything to that?
No, I agree very much with the point. It's the kind of activity that we do with our partners in the NHS trusts. Indeed, on this exact topic, we had a so-called Café Scientifique just earlier this month. It's important to get these concepts across. The one community that we actually haven't spoken about in terms of the need for education is the GPs, because I think this is highly specialized knowledge, rapidly evolving knowledge, and finding the right mechanisms to educate them as well is as much as of a challenge, and sometimes more than not knowing how to reach the patients.
I mean, you're not getting much help really from the press, are you? I mean, I think as Professor Barker said, it's either a disaster or a triumph, so to speak. And the amount that you can do as individuals is quite limited. You can talk to specific audiences. But surely the GPs are the ones who see patients all the
That's
time.
exactly the limitation I would point to in our activity of last week. If it was 40 patients or 50 patients or 60 patients, it's just a drop in the bucket. It's a worthwhile activity. Reaching the GPs would be important.
It's the headline writer. That's who you really need to educate. But of course, their job is to sell copy, not to study the true nature of the story. Exactly. Because the articles themselves are often very accurate. It's the headline that's misleading. And that's, of course, what the patient takes away.
Of course.
I suppose, if I may say, one of the opportunities is modern media. I think we've already explored how certain parts of the media, by necessity or whatever way you want to define it, have to tell stories in a much more extreme way. But I think with increasing democratization of the media through the internet and Twitter and various other things, that does become an opportunity to present a more balanced view, at least that to the public, that they can access. I think the issue is availability. I think one of the great difficulties of modern life is, where do you look? You get covered in an avalanche of data, and the question is, what makes somebody go to one source and the other? And I think if we can resolve that, I mean, there's a beautiful section on Eurostem cells, on stem cells for eyes, a nice little simple article that refers to all the current technologies. But the problem is, how does the public get there? What leads them there? What do they trust as their source of information? And I think if we can help that guidance for the public, and also make them want to go there through other reasons, then that can help a lot.
Okay, I'd like to turn to Lord Patel.
Thank you, Chairman. My question from the Twitter relates to the impact that the European Court of Justice ruling in Brussels versus Greenpeace case would have, and in particular the investment in research, and investment in research particularly from the huge European funding that's available for research. Would you like to comment?
I think it's worthwhile turning back to Lord Winston's question about iPS cells versus ES cells to put this one in perspective. Although iPS cells in principle and in substance can adopt many, if not all, cell fates, both as a platform for regenerative medicine and as a platform for related activities like pathway dissection, drug discovery, and drug safety testing, the question isn't whether the cells can become a neuron or can become a heart muscle cell or can become a blood-forming cell. The question is just how good are their functional properties once they've made that lineage decision. And one of the most important and instructive examples that I've seen recently on the need for continued work in human embryonic stem cells was shown at a drug discovery symposium in Manchester last month, where an investigator from industry, from AstraZeneca as it happens, was doing systematic comparisons of drug toxicity in a panel of human ES cells on the one hand and human IPS cells on the other hand, using cardiac muscle made from these different sources, and found, for reasons that are not yet understood in a fundamental or reductionist way, that the findings in terms of predictions of toxicity were very, very strong using cardiac muscle made from embryonic stem cells, and weaker, much weaker, using the cardiac myocytes made from IPS cells. That's not to say that those kinds of problems won't be sorted out in the future, but I just point to that as an example for now where the ES cells would clearly be out in front. It seems to me reasonable that a company like AstraZeneca or others ought to be able to engineer the ES cell, human ES cells, in different ways that make them particularly well suited for the kinds of toxicology studies that they need, for the kinds of target discovery activities that they need to come up with new drugs. But currently, if they were to do that in the cell product that's best for the purpose, they wouldn't be able to patent it. If they were to do the same manipulation in an iPS-derived heart muscle cell, less fit for purpose, they would be able to patent it. And so I think that's one example at the drug discovery and big pharma level where the EU court decision is very destructive, putting aside all issues relating to human ES cell-derived heart muscle as a potential therapeutic product, where it would seem logical that the steps involved in bioprocessing, in ensuring genome stability in long-term processed cells, in purifying the cells to homogeneity, and also to delivering them, all of that would seem to qualify for intellectual property protection were it not for the latent religious objections to embryonic stem cells on the part of a small but vocal minority.
Okay, are there any other brief additional comments? Because I'd like to come to one last question from Lord Wade in a moment.
Just to comment that actually, of course it's uncertain, but certainly the partners that we're working with, our group, our centres working with, which is Pfizer, have been surprisingly robust about moving forward despite the ruling, which is encouraging. I think given that sometimes the problem is also an opportunity, I think going back to you know, what we heard just now about the engineering issue, I think modern stem cell technology is often a complex interplay of some of the high-level technological manufacturing issues, delivery issues. For instance, we've had to design special instrumentation to deliver some of the stem cells that we're doing so. So I think if we're clever about it, let's put it that way, and work in a multidisciplinary way, there can still be the platform for investment with some protection for the companies who invest. And again, that belies the issue of the technological investment that the UK makes into this field. It's not just doing the cell and all the biology behind it. There's a whole raft of technological co-patenting that occurs that makes it a unique product. So I think if the UK were also to invest in some of the co-investment, then actually the value would still be there, and I'd just like to make that point.
Yeah. So for a final question, Lord Wade.
Thank you. The Twitter question is, would possible long-term benefits to society justify increasing tax breaks for promising research for regenerative medicine? I suppose the underlying question is, what are the long-term funding implications for the development of this technology?
Well, that's a tricky one, isn't it?
I don't know the answer to. I mean, it's such a complicated area because there's so many ifs and buts and what happens really. And at the moment, I'm just happen to be involved with a group of scientists where this is a big issue with the withdrawal of one drug for one indication because they've suddenly found it to be very successful and it coming back at a higher price because they now know it works. So it's always this issue of, you know, once you found that something works, how do you take it out and offer it in a sort of favourable way to as many people as possible? And I don't know the answer to that. I think if I speak for myself in terms of the world of neurology, I think these therapies will never become mainline therapies for a vast majority of people. I think they will always be kept for specialist centres for relatively select groups of patients. And I think they will therefore get the funding they need in order to have that therapy, because it will be a restricted group. I think it becomes different when you move it out of, say, the field of Parkinson's disease and you have a treatment that could help everyone with diabetes or prevent them getting diabetes, because suddenly you have a vast market, and how you actually can regulate and support that, I don't know.
Any comment from Professor Kaur or Professor Schneider? Professor Schneider, perhaps.
I think the need for more funding is there, but I leave it to others wiser than myself to decide whether the financial instrument should be tax breaks versus grants and other kinds of mechanisms, such as support through the Technology Strategy Board.
I mean, I feel very strongly, if you look at what we've been successful with in getting through to First in Man and some success, it's relied on a variety of things, but if you look at the way the funding packages were put together for these highly successful first or second in the world type initiatives that we do, it's very diverse. It's been a lot of leadership from the people who head up these programs, who go all over the place to find the mixture of funding that you need to make this, without which these initiatives, I wouldn't be sitting here talking about, you know, our centre having these first in the world things for the UK. And there's no question that the partnership with the commercial sector is essential. Pete Coffey makes the point that so far, for the London project, which is advancing rapidly, which is a British-based thing, which we'd have to put huge infrastructure in, both from charities, from MRC, from California, from a private US philanthropic donor, that, you know, he's had to fill in, they've had to do 37,000 pages of documentation so far. Now, to do that sort of, you know, no academic on the planet can do that. Even though they have all the ideas and the science, they need support. They need support probably from specialists in the field who tend to be located in industry. Industry needs to have some incentive to come in and work with us to write 37,000 pages of documentation. So whether it's tax breaks or other thing, I think what is absolutely essential is there has to be support for this intermediary funding, without which we just simply cannot take these treatments through to clinical usage.
Thank you very much. I think we need to draw... Sorry, we do need to...
Wash out, go on. Okay.
Sorry, is it very, very brief?
Well,
running over time.
Is this an issue you think that we should look at in this report?
Absolutely, because I think it's essential for success. If I look back to the things we are succeeding in, without that degree of support to fill in this huge amount of you know, regulation and administration that you need to get through this page, we cannot possibly succeed in bringing treatments through, particularly in the UK, for UK-born ideas and technology through to our patients without this help, either from industry or elsewhere.
Thank you.
Okay, thank you very much. And I'm sorry we've run slightly over time, but it's partly a reflection of the very interesting answers you've given to our questions. It's been an excellent session. Also, I'd like to thank all of those who sent in the Twitter feed questions. We didn't get through all of them, but I think we've got at least a response to some of them, and others were covered implicitly in the earlier responses. You will, in due course, receive a draft of the transcript for you to make any corrections. If there are any comments that you want to make in addition to those that you've made today, do feel free to write in, and it will be included in the evidence that we publish with the report. But thank you very much indeed for your comments today.
Thank you.